Okay, so we'll begin now. Hello.
Okay, Dr. Khaled.
Okay, welcome to the third session of molecular and genetics application. This is Dr. Khaled Sabarna from Palestinian Al-Ahliya University in Bethlehem. Our next speaker will be Dr. Nehal Abo, Department of Biochemical Engineering and Chemical Engineering on Texas University, Texas. Dr. Nehal, the mic is yours.
All right. Can you see my screen?
Yes, we can.
Okay, perfect. So thank you for everyone, for all the organizers, for thinking of me for this really nice opportunity and to get to meet as many of you. Thanks specifically for Omar for the invitation, for Sharaf and Ayman for all the details and logistics for the meeting, and it's really my pleasure to be here and to be sharing some of our research with all of you in Palestine. All right, so today we are going to be speaking about the effects of nutraceuticals on our abilities to engineer articular cartilage tissues. This is a collaborative project between many institutions, and I'm just speaking about the net result of that project, of one study from that project. Let's start with articular cartilage. This is basically the specialized tissue that wraps all moving joints. So think about spine, knees, fingers. It has two main functions. The main function is for it to be a lubricant when the bones are rubbing each other. And also in some joints, like in the knee, it's a load-bearing substrate. It's about 2 to 4 millimeter thick. As a tissue, it has the least amount of cells of all tissues in the body. And the cells are surrounded in the tissue with an extracellular matrix that is heavily based in collagen as well as glycosaminoglycans or proteoglycans. This tissue is problematic because it doesn't have any blood supplies or lymph nodes or nerves. And so basically upon injury, there are no blood supply to the cells in order to regenerate the tissue. In addition to that, it's subject to very harsh chemical environments, including hypoxia, acidity, and sometimes high osmotic pressures. The tissue itself, if you look at it, is also heterogeneous. It's multiphasic, has solids and water in it. It has 4 different zones, and these zones vary in everything you can think of. They vary in the cell density, in the protein composition, in their porosity, in their mechanical properties, in the alignments of proteins within them, and as well in their function. And so you can start to envision that if something happens to this tissue, engineering something that looks representative of this is a hard task in the lab. When the tissue is damaged, we get osteoarthritis, which I'm going to refer to as OA. OA is basically has many risk factors. Of course, one of it is a trauma, but also aging due to wear and tear, the use of the joints, as well as it's associated with some diseases like diabetes and some infections. It's more prevalent in females than males, and also obesity as we put more load on our joints, and sometimes heredity. About 30% of the population above 45 has osteoarthritis, 80% above the age of 65 has osteoarthritis, and every one of us will have osteoarthritis at some point in their life. As a disease, this is a whole joint disease. It's not just like an articular cartilage degradation. If you think about any joint right here, you can see that some of the factors associated with this disease are going to include the degradation of the cartilage, muscle atrophy, osteophycation of bones, as well as synovial inflammation and swelling of the joint. All these factors will allow the people with osteoarthritis to have either hard bony enlargements, a lot of pain, and oftentimes the disease will end up with a disability. Osteoarthritis is the leading cause of disability reported world-class worldwide. All right. Currently, there are no disease-modifying drugs to osteoarthritis. That means we cannot treat osteoarthritis. And the only way to deal with it is to manage it. And managements come in physical ways, like losing weight, physical therapy, having some devices and braces to assist with movement, and sometimes chemical treatments like taking anti-inflammatory, drugs or pain-relieving drugs, or sometimes injections like hyaluronic acid or steroid injections, and sometimes comparative, alternative medicine like the point of the talk today. But eventually, we will need to go to a knee replacement or a joint replacement, hips, for example. In the US, every minute, there is a total knee replacement, okay? So this is significant invasive surgery that calls for alternative for us to find ways to manage osteoarthritis. The onset of the disease is only going to continue to worsen as the obesity and as well aging is improved with time. So the alternatives come with tissue engineering. And tissue engineering as a field in general aims at engineering the tissue in the lab and then hopefully personalize that tissue and take it and implant it in the body in replacement of the damaged tissue. Here we are talking about articular cartilage specifically in hopes to restore the function. Tissue engineering has many components. The first component, of course, cells. You have to start with a cell source in order for the cells to start to proliferate, express an extracellular matrix, and form a tissue. The cells usually are sitting on a scaffold that is mimetic of the in vivo scaffolds to the extent possible. You feed cells with growth factors or nutrients, and then you put everything in a bioreactor that really represents your microenvironment. And then you wait for some time in hopes for the cells to do the job for you and give you a functional tissue. Now that is really easy said than done. You can envision the interplay between these many different, factors that need to be optimized. And this is why up to now, we do not have a functional articular cartilage tissue on the market. All right, in our group, our long-term goal is to investigate a variety of questions when it comes to all these four components, like the mechanical, chemical, and physical environments and stimuli that are applied to cells. We want to design unique bioreactors that really represent the microenvironment and engineer a functional tissue to the extent possible. For the sake of time, this talk right here is going to focus only on one question that we asked, and that is to investigate the effects of nutraceuticals on the abilities of osteoarthritic chondrocytes extracted from donors of variable age, weight, and sex to promote the performance of a mature extracellular matrix and reduce inflammation. Our hypothesis is that if we add these nutraceuticals to the monolayer cultures of cells, the cell's ability to produce the extracellular matrix are going to improve, as well as the reduction of inflammation is going to be obvious. And all these effects are going to be dependent on the age, sex, and weight of the donor. So what are nutraceuticals? Nutraceuticals, as the name implies, are materials that provide nutrition, but also they have a pharmaceutical value to them. And nutraceuticals are taken by 47% of osteoarthritic patients in hopes for help in relieving of symptoms. The four that we investigated in this study are gallic acid, which we can find in a lot of fruits as well as in some nuts, alpha-tocopherol or vitamin E, which is available in mixed nuts and broccoli and olives, catechin hydrate, which is the green tea extract, and ascorbic acid, which is vitamin C. So we looked at these four specifically because they are antioxidant and also they are anti-inflammatory. And we wanted to see if we can have an effect on engineering articular cartilage. In order for us to do our experiments, we went ahead and worked with an orthopedic surgeon and took articular cartilage specimens from patients who underwent total knee replacement surgeries. We worked with 25 donors in this particular study. Once we received the tissue, we looked at the least inflamed part of the tissue, digested the tissue with collagenase 2, and then obtained the cells out of the tissue. The cells that are residing in articular cartilage are called chondrocytes. These are the specific type of cells we are interested in. Oftentimes, we will get 1 million cells per knee that is donated. And so that's a very limited number of cells to work with. So for this particular study, we had to wait a year in order to collect enough samples and enough donors to perform the study that I'm sharing. All right, after that year, we divided the cells that we received into two groups. The first study that we are talking about was a study for females only. So we fixed the cells, the sex, and then the age and weight of these groups were different. Group 1 was females between 50 to 69 years old, and the average weight was 100 kilograms. And so we are going to call that the younger and heavier group. And group 2 is the 70 to 80 years old with an average weight of 75 kilograms. So this group is the leaner and elderly females. And we wanted to see how these two groups of females respond, cells from them respond to the nutraceuticals. In our second study, we are investigating the effect of sex. So here, the weight was constant, the age was constant, about 65 years old, but then we have a group that has male and a group that is females, and we extracted cells from them. So let's go ahead and take a look at how we approach this experiment. So we went ahead to the lab. We take the cells, right? We reserve them until we need to use them, and then take them, sheeted them on tissue culture plastic until they formed a confluent monolayer of cells, and that become, our culture that we are going to work with. Our negative control was the culture without any treatment, so the cells just were fed a growth media that will make the cells heavy. Then we have our positive controls, which were addition of a nutraceutical at a 50 micromolar to the media. And then after we let the cells grow for 21 days and optimized and looked at the cell performance to form the articular cartilage tissue. So I'm going to share our results with how the cells performed in the lab, but for the sake of time, I'm just going to show examples of the results, not all comprehensive results of the two studies. The first thing in mind to prove is viability. Are these nutraceuticals cytotoxic? Are they going to kill the cells or not? And so it's very important whenever we use something for it to be safe to the cells. And so what you can see here from the sixth study, that when you compare the viability compared to the negative control, so no addition of a nutraceutical versus added nutraceuticals, you will see that we are about 100% for all of them. Sometimes we are improving the viability of cells. And so the takeaway message here, our nutraceuticals were not cytotoxic. And this was evident the same way for our age and weight study as well. So with that, we are good. We move now to thinking, did we provide an extracellular matrix that is key or representative of what we find of articular cartilage? And so the two main components of an extracellular matrix in articular cartilage are collagen and glycosaminoglycans. So our first measure of those was staining. And so we stained for collagen, and you see, irrespective of the treatment that we have for nutraceuticals, we always express collagen in all tissues. This is qualitative, by no means it's anything, but it's just telling us that we have.
لما تطلع تطلع الله يسلمك يعني اكثر واحده فينا لما تطلع
حياك الله.
Can you guys mute?
دكتور خالد كان مفتوح المايك تاعك.
Okay.
Sorry,
So
تفضلي.
no worries. So we express collagen in all these basically tissues irrespective of the treatment. A similar way, we wanted also to confirm that we are expressing GAGs or glycosaminoglycans, and indeed we have them expressed in all our tissues at the 21 days. So this was good, but it's a qualitative. So we were looking for a quantitative way to think how much really I produce of collagen and GAGs. So to do that, we did what we call assays. So basically, you apply certain dyes and then you measure your outcome using spectrophotometry. And so what we see to the left, for example, the males and females, that in all the treatments, we have collagen, that's number one. But the second thing that I would like you to notice is that males are always producing more collagen than females across all treatments, even for the negative control. If we go, for example, for the GAGs, again, we produce GAGs in all treatments, and also males produce more GAGs than females, except for when we have used ascorbic acid. Sometimes I'm going to show stars and sometimes we are not showing stars, so that to donate basically if we have a statistical significance or not, and it's very hard to just keep memorizing if we were statistically different or not, but I'm going to conclude everything together later on to show what worked, what did not work from all these nutraceuticals. Here the point is the example of how these results were collected. Another important thing that we wanted to look at is inflammation. Remember, this is a joint disease, and the use of a nutraceutical will be great if we can reduce the inflammation in the tissue. And so we brought for nitric oxide content, which is an indication or a measure of inflammation. We did that on day 1, and then we did that on day 21. So day 1 was after congruency and before we did any treatment. And so you can see that cells were more or less at 8 micromolar nitric oxide at day 1, whether they are females or males. And then females were slightly higher than males. And then at day 21, we reduced inflammation for both groups, males and females. This was a big thing for this study, that the nutraceuticals are going to reduce the inflammatory levels in these tissues. All right, another important thing that we looked at are the mRNA gene expressions, okay? So we were really interested in looking at the mechanistic details of what is going on in these 2 studies. And we looked at 4 different categories of markers. The first one is what we call as chondrogenic markers. By the way, anything you see in green here is something we want more of. Anything you see in red is something we want to decrease. So the chondrogenic markers, these are the things that are desired phenotype for the cartilage, such as collagen 2, collagen 11, and aggrecan. So you want more of those. Hypertrophic markers or osteogenic markers are collagen 10. We do not want to have hypertrophy or de-differentiation of cells. Then we have some regulatory markers, such as the polymorphogenic protein degradation pathway. We want that to be less. Some anti-inflammatory, pathways like upregulated by FOXO1 that improves autophagy, and then SOX9, which is a key marker for the early stages of chondrocytes proliferation. We want more of. We want less of the nitric oxide synthase, which is an indication of inflammation, MMP13, which is an indication of degradation, and we want more of TNF-AIP6, which is an anti-inflammatory marker. So let's go ahead and see. We measured all these for all the treatments, for all the studies. An example of what you get for one marker for one study is going to look like the histogram we see right here. And this is for nitric oxide synthase. And you see that is shown for group 1, which is the younger, heavier females. Group 2 is the older and leaner females. And we measure that as a function of the treatment, like negative control or oxygen hydrate or gallic acid or vitamin E or vitamin C. One thing that you see here, for example, that group 2, which is the older and leaner, produced a lot less from this NOS. So they really did well compared to group 1 because we want to decrease this marker. Remember, it's an inflammatory marker. So we have histograms for all these different markers, and then we are gonna basically look at how they behave and think about all our studies combined. All right, so let's go ahead and discuss the first study. The first study was the age and weight. Our group 1 was the age 50 to 69, 100 kilograms, so younger, heavier. Group 2 was our elderly and leaner females. These are all the markers that we looked at. Anything boxed in green, we want higher. Anything boxed in red, we want lower. So let's take a look. You can see as when it comes to collagen 2, the elderly females produce a lot more than the younger females. This is a phenotype marker, like it's desired in articular cartilage. When we look at aggrecan, they were similar. And when we looked at SOX9 and FOXO1, group 1 produced more. Now, and they produce more of the TNF-AIP6, which is an anti-inflammatory. Now, if you look at the red ones, these are the ones we want less. By all means, group 1, which is the younger and heavier females, produce more of the worst markers than the elderly and leaner females, which was counterintuitive. Like you would think younger persons would do better, you know, and will be able to respond to the treatments better. This was not the case. If we think about how all these markers interplay, FOXO1, if we produce more of it, we produce more of SOX9, and that promotes aggrecan and collagen 2 production, which is what we want. If we think about the degradation protein BMP2 and the inflammatory environment NOS2, having more of these will bring MMP13, which is a degradation of cartilage, that will block the formation of the desired aggrecan and collagen 2. With all that in mind, this study pointed to the fact that chondrocytes of the elder and leaner females were less inflammatory, and they had a very similar phenotype to the younger females and more obese. This finding really stressed weight management. Like the only thing that we can think about as bad in this younger group is obesity. And so obesity should be studied individually when it comes to osteoarthritis. If we look at the second study, which was comparing the males and female responses, this table I'll walk you through. Let's take a look at the top half of the table. These are all the bad markers. These are the, you know, things that we want reduction of. And if you look at the second part of the table, these are the things that we want more of. So if we look at the top half, we want our arrows to be all red, pointing down, okay, reduced compared to our negative control. And so if you look at the females right here to the left, you can see that more or less all nutraceuticals were similar, except gallic acid has 3 red arrows. That's slightly better. So maybe females need to take more of gallic acid to decrease the anti-inflammatory environment. While if you look at males, alpha-tocopherol outperformed all nutraceuticals. So vitamin E is going to reduce the effects of the inflammatory environment in the male cultures. If we look at the performance when it came to enhancing the articular cartilage phenotype, we want to see more green arrows at the bottom. And so you can see for both females and males, alpha-tocopherol was important to reduce or enhance the extracellular matrix formation. So with that, in females, we observed that the gallic acid reduced inflammation, while alpha-tocopherol or vitamin E enhanced the ECM formation. In males, alpha-tocopherol reduced inflammation and enhanced extracellular matrix formation. And this study results suggest that the effects are sex-dependent. One interesting thing that we observed that even though like just feeding the cells with a negative control, which is just like a growth media, was sufficient to decrease inflammation, even if you did not use any nutraceutical. So that's a takeaway also, because like in actual in vivo conditions, the cartilage is deprived of nutrients. Also, all our cells were viable, so the nutraceuticals were not cytotoxic. Nutraceutical use in basically by the public is not controlled by FDA or any agency. A nutraceutical does not have to be clinically approved to go on the market. It's up to the person to use it or not. And so at least they are not cytotoxic. So one can use them, and then if they don't benefit, they stop using them, and that should be okay. For the future, we really should increase the number of repeats for our experiments. We should investigate responses from individualized donors. We should investigate other phenotypes like diabetes, like obesity on its own, irrespective, independent of age. Look at infections, look at different populations. We should culture cells in 3D matrices, not just in mono layers. We should also look at how can we engineer a disease model for osteoarthritis that prevents us from the need to collect tissues from diseased personnel. Because it took us a year to just collect cells for this study. And osteoarthritic cells are already stressed, and so they don't perform really well. And so if we can make a disease model that we can use for screening of the drugs, that can be a big improvement in the field. And with that, I would like to thank the students who did this work, the 3 PhDs, Haneen, Alia, and Mahmoud, and funding for this study from National Science Foundation and National Institutes of Health, as well as our industrial collaborator, Regeneron, for performance of mRNA studies, and our orthopedic surgeon for providing all the tissues. And thank you for listening, and I'll be happy to take your questions.
Thank you very much, Dr. Nehal, for this very interesting and promising studies. And I hope that the audience will have any questions. Please, could you please just any questions for Dr. Nehal?
I think there has been a question on the chat, Dr.
All right, let me take a look.
Were there any intra-group variations?
Yes, so if you are referring to between a nutraceutical to nutraceutical and a nutraceutical to negative control, in the age and weight study, okay, where there were females, there were no intra-group variations. So all in nutraceuticals performed more or less the same. But when it came to the sex study, a study with males and females, we have observed very limited intragroup variations with alpha-tocopherol and ascorbic acid and gallic acid, sometimes, you know, optimizing a factor versus the other. The main takeaway from here is really nutraceuticals did not really perform significantly compared to a negative control, just media that you provide to cells. That means because cells in native tissues are so deprived of anything, even providing the nutrients was sufficient for them to do well. The nutraceuticals were really good in reducing the inflammation. Now, this is important because this is a joint disease that has inflammation associated with it. If we decrease inflammation, we decrease like MMP13 degradation, and we postpone the progression of the disease. So our study really did not find so much benefits when it comes to forming articular cartilage for use of nutraceuticals. You can think of it as maybe a negative result in that manner. Like, I wish I can say, oh, all of you go ahead and take gallic acid or take this. No, that was not the case, you know. But taking these nutraceuticals was not cytotoxic. It was not harming the cells. It reduced the inflammation, and it's something we can take off the market, while a growth media that you go is not something you find on the market. So they perform similar to the growth media, if that answers your question.
Again, thank you, Dr. Nehal. If anyone wants to raise his hand or any questions.
Dr. Omar.
Me.
Yes.
دكتور عمر كمان رافع ايده.
Yes, okay. مش شايف اللي تفضل دكتور عمر.
Good afternoon, everybody. How are you, Nehal?
Good to see you.
Thank you very much. We are glad to receive you here virtually.
Yes.
Thank you for accepting our invitation, actually, to join with this fruitful work and amazing guys, I can say.
My pleasure.
My questions is all the time related to safety and how the system is safe especially for donors, I mean, to avoid any diseases or something like this.
You mean once we engineer the articular cartilage and put it back as an implant, how this will be safe? Yes. So this is a very good question. And first, we are so far from that, okay, application. But you have to think about where are the non-safe elements are going to come to this implanted tissue. We hope for it to be personalized. So hopefully the cells are coming from the patient and they are used in the lab to grow the tissue and putting back in the patient. So no immunogenicity reactions or rejections from that standpoint, because hopefully it's personalized. Second thing that you have to worry about is the scaffold. Like you have to grow these cells on a scaffold material. And right now we have scaffolds that are synthetic and scaffolds that are biological. And we have to understand the fate of the scaffold in vivo. And so this is going to be a very important question. How cytotoxic is the scaffold? What's the degradation rate of the scaffold? How long will it take for the scaffold to degrade in vivo? Where are the outcomes going to go and disappear in all organs in the body? And this is why, for example, in our lab, we are interested in hydrogel components for scaffolds that are native to the articular cartilage. So like our scaffolds are hyaluronic acid-based scaffolds that are native to the tissue itself. So they are not foreign. But that's an important thing to ask as well. And then you have to consider infections basically associated with surgeries. These are open surgeries. You are putting the implant back. Anytime we open a tissue, we have the risk of an infection. So there are a lot of questions with that regard. And a major one also, can we really grow a large size articular cartilage in vitro that fills the entire defect in vivo or not? And so all these questions are open for investigation. I wish I have an answer for you, but there is not. People are working on all these things to be considered.
Yeah, thank you. Thank you very much.
You're welcome.
Okay. Thank you, Dr. Nehal, again. And we'd like to move to the second talk by Sondos Odeh and the other two colleagues, Mahmoud Zahaika and Zaydoun Salah from the Arab American University, titled by Functional Role of TET3 Enzymes in Breast Tumorigenesis. Okay, it's yours. The mic is yours.
Hello, can you hear me?
Yes, we can.
I hope everyone is doing well. I would like to thank you for the opportunity to present my work today, titled Functional Role of TET3 Enzyme in Breast Tumorigenesis, supervised by Dr. Zaydoun Salah. A special thanks to him, of course.
Okay.
So first of all, I would like to start talking about cancer, which is a complex disease, and it is characterized by rapid and uncontrolled cell proliferation in a tissue. This feature of uncontrolled cell proliferation affects the normal levels of gene expression, which in turn also affect or have a manipulative role on normal cell function. And of course, normal gene expression correlates directly with normal cell function. Cancers are thought to be genetic diseases. They arise of sequential alterations. These alterations can cause oncogenes to be activated and tumor suppressor genes to be inactivated. Now, according to the WHO, breast cancer has the highest morbidity and mortality rates among all cancers in women. Breast cancer is said to be heterogeneous, meaning that there could be multiple subtypes of tissues that can comprise one tumor. It is said to manifest different malignancies in the mammary gland. For some time now, scientists have thought that cancer is from genetic events such as mutation, which can eventually cause the initiation of cancer. However, recent studies for the past, let's say, 2 to 3 decades have been emerging, suggesting that there is a role for epigenetic modifications that take place in tumor initiation and progression. These modifications are multiple. They are responsible for different processes and different cellular functions in a tissue. For example, we have DNA repair. They are responsible for replication and so on. One of the very important DNA modifications, which is our main focus here today or in our study, is the DNA methylation. It is an important type of modification. It is thought to be irreversible and heritable state. It is catalyzed and maintained by DNMTs or DNA methyltransferases. It is, of course, the addition of a methyl group to cytosine. It is responsible for cell proliferation, apoptosis, differentiation, and other processes. Regulation of DNA methylation should be very important because aberrant DNA methylation can result in emerging of cancer and by hypo or hyper methylation. Methylation, as I said, is very important, and we have emerging research that indicates that there are some transcription factors or some related proteins that do not only protect sequences from methylation, but they also initiate DNA demethylation. And contrary to DNA methylation, demethylation is defined as the process of removal of the methyl group from the cytosines. This can be passive or active. Passive DNA demethylation has been studied and it is known for scientists. However, active DNA demethylation was elusive until the discovery of the TET family of enzymes. The TET family of enzymes, or 1011 translocation enzymes, are a family of 3 enzymes, which are TET1, 2, and 3. They convert 5-methylcytosine to 5-hydroxymethylcytosine, which can be further converted to unmethylated cytosine. These enzymes, the 3 of them, are differentially expressed in several tissues and differentially expressed in different cancer types. Our main focus in this study was TET3. Studies have shown that the role of TET3 is different from one cancer type to another and from one tissue type to another. It is predominantly expressed in oocytes, zygotes, and neurons. TET1 and 2 have been heavily studied by scientists. However, TET3, there are very least studies about TET3, and so, which brings us to our study aims. We aim to investigate the expression pattern and functional role of TET3 in breast cancer, and to elucidate the function, the effect of TET3 overexpression in breast cancer cell lines in different cellular contexts. I will be brief here for the sake of time. So first of all, to study the function of TET3, we had to produce cells that are overexpressing of this gene. We started with cloning the target gene, which is TET3, into a lentiviral vector, then choosing the suitable cell line, of course. So we tested multiple breast cancer cell lines, but we found that the TET3 expression level was the lowest in the MD231 cell line. So it was our cell line of choice, simply because we have a room of overexpressing the gene. Then we moved to the steps of transfiction and infecting our chosen cells with the target gene. We also infected the chosen cells with empty vector just to serve as a control. After that, we validated the results by testing the expression of TET3 in the infected cells with the target gene compared, of course, to the empty vector. And we indeed found that there is an overexpression in the infected subjects. After that, we had to test the effect of TET3 overexpression on the cell phenotype, MDA and B231 cell phenotypes. So we studied different hallmarks of cancer. These included cell proliferation, motility, and cell migration, and cell survival. First, we did the XTT cell proliferation assay, and we found that TET3 overexpressing cells result in an increase in cell proliferation over time, of course. Then we did the wound healing assay. We found that TET3 overexpressing cells result in a higher rate of cell migration, meaning that cells infected with the target gene move in a faster and higher rate compared to the empty ones, and that they closed the wound. After that, we used the cell survival assay, and we tested. We found that TET3 overexpressing cells result in an increase in cell survival. Overall, our results indicate that TET3 here exhibits an oncogenic behavior. Then we moved to to find the relation between the phenotypes that we found to the genotype. So we tested the effect of TET3 overexpression on other genes like oncogenes and tumor suppressor genes. Interestingly, here we found that there is a differential effect on these on the studied genes. Some of them were elevated, while others were downregulated. But some oncogenes showed higher expression, which indicates, or which also supports our results, in that TET3 is in fact an oncogene. Then we tested the TET3 enzyme overexpression on breast cancer tissue samples taken from Palestinian patients. We found that there is differential localization of TET3 in the breast cancer tissue, and it depends also on the stage of cancer. For example, in DCIS, which is ductal carcinoma in situ, which is a very early stage of breast cancer, TET3 was found to be downregulated, while in invasive forms of breast cancer, it was shown to be highly expressed, which also indicates that that TET3 is an oncogene. Lastly, we wanted to correlate our results with clinical outcomes using the Kaplan-Meier online tool. We tested, we found that high TET3 expression predicts poor prognosis in breast cancer. All the patients presented here, or all the figures presented here, are representative of patients with breast cancer, showing poor prognosis and less months to live compared to those with a lower level of TET3 expression. This brings us to the conclusion. One of the conclusions was that different expression patterns of TET3 are present in different cancer tissues and in different our results show that TET3 has an oncogenic behavior, at least in the context and the specific breast cancer types that we studied. We also found or speculate that there are other factors that need to be further studied. These factors play an important role that needs to be elucidated more. For example, it was found that TET3 has at least 3 isoforms. These isoforms are expected to play an important role in the role and function of, or roles actually, and function of TET3 in breast tumorigenesis. And now for future studies, we aim to clone the different isoforms of TET3 in order to have a better and wider vision of the function and behavior of TET3. We also aim to study the function of TET3 by knocking out, for example, one of the isoforms or the different isoforms to see just the effect of on the cellular phenotypes. And thank you.
Thank you very much, Sondos.
Thank you.
I'd like to ask the colleagues if they have any questions, please. Hashim raised hand. Welcome, Hashim. Hashim.
Hello, Jamia. Just a quick question. You showed the women with the breast cancer that had a higher TET expression level to be of bad prognosis, right? That was in the last 2 slides, I think.
Yes, this was an immunohistochemistry experiment.
No, the graph.
Oh, the Kaplan.
Yeah, exactly. So my question here, do you know the causative gene, the mutations, in which genes that cause this cancer?
I'm sorry, can you repeat the question about that?
Is it BRCA1, BRCA2, is it P3? Which gene is mutated in here?
Here we only studied we knew that these cancer patients had higher level of TET3. So,
but you don't know which mutations do they have, if they had.
the Kaplan-Meier tool allows you to have different parameters when you want to show a result. So some of them might have triple negative, for example, breast cancer, or different grades, for example, but not exact genes. So it's just different parameters generally.
Yeah, because you're talking about epigenetics.
Yes.
And I don't know, I'm missing a point here because you have so many different causes for breast cancer, both on a gene level and also on an epigenetic level. And now you're trying to overlay a third parameter, which is the TET overexpression. I don't know if you're getting my point here. I'm missing some information, whether the TET overexpression overrules whatever mutation that are found on those cancers.
We didn't go that deep into that. We only studied the overexpression of TET3. If it has any other correlations or effects on different other genes or other factors, we didn't go that deep. We only wanted to prove the overexpression of TET3, just because we know about TET1 and TET2 and how sometimes if they are overexpressed, they have, for example, TET1 was found in some tissues to have an oncogenic behavior, while in others it was had tumor suppressor gene. So we just wanted to show the result of overexpressing TET3. That could be for further studies to see the relation.
Okay, thank you.
Okay.
Dr. Khaled.
Yes. Thank you, Sondos, Odeh, and your colleagues. And for the sixth time, we'll go directly to Sabha, Rabaya with Hisham.
Please stop sharing. No, stop sharing your presentation.
I am trying.
Yeah, there is a sign, red sign in front of you.
من عندك يا دكتور وخلاص.
Okay. Okay.
كان الدكتور عمر رافع ايده، بعرف مش دكتور عمر كوميرشال كان رافع ايده.
يعني اذا في اسئله خليها للجنرال ديسكشن بيكون احسن.
It doesn't matter. We can ask later on. Thank you.
تمام دكتور تمام.
Okay. As I said before, Sabha, Rabaya and Hisham Darwish from Arabic American University, Palestine, Ramallah, Palestine. Their talk will be about the correlation of major iron metabolism related genes, single polymorphism with autism spectrum disorders in Palestine. Okay, welcome, Sabha. Sabha or Hisham Darwish, are they here?
صبحة كانت موجودة، صبحة قبل شوي دخلناها، وينها؟
Yes. هاي صبحة دخلت. Okay, صبحة. Could you please share your screen? صبحة. Okay, Sabha, can you hear me?
Yes, Doctor, I can hear you.
Yeah, you can share your screen and begin your talk, please.
Okay.
Do you have a problem in your?
I think, yeah, there is a small technical problem. I don't know if it appears.
Okay, you are about to do it.
Yeah.
Yeah.
هاي اشتغل على ما يبدو بس.
Rabaya started screen sharing. Yeah, you can click the file now, please, and go ahead.
Okay. So you see the slides?
No, not yet.
Okay. I don't know what is the problem.
The problem is that we cannot see your file on PowerPoint presentation. We have a message says that Rabaya has started the screen sharing, but actually we cannot see any file. Do you have anyone next to you who could help in sharing your screen?
Yeah, sometimes it's in Zoom settings. So maybe for the sake of time, if someone else has her PowerPoint and they can share, or if she can email her PowerPoint.
You prefer to skip your time. Maybe you will be ready after Dahlia Abu Hlayl. Could you please stop share your screen? Stop, please.
Okay.
Thank you.
Just give me a minute, please.
Yes. Okay. Sabha?
Yes, I'm trying to stop sharing, but I don't know what is the problem. It is.
ملف وقف لانه انا وقفته من عندي يا دكتور.
اوكي.
يعني تقدر تعمل نيكست سبيكر. شوف الثاني.
Dr. Sharaf, do you prepare to go to the next presentation until Sabha will be ready?
Yes, go.
Okay. Okay, Sabha, you may solve the problem, and after a quarter an hour we'll come back to contact you.
Bisan
No,
can start now. Bisan.
Dahlia, Sharaf. It's Dahlia.
Bisan. نعم, the next one.
Okay, Bisan al-Fasfoods by directional PCR amplification of specific alleles. PASA for of Borola gene cytosine Palestine. Bisan, are you ready? I even cannot see her on the panel.
Hi, Odeh. طيب ماشي.
Dahlia is there.
Okay.
Yes, doctor, I'm here. I can start now.
Okay, Dahlia, we'll save the time.
بيسان برضه صارت موجودة دكتور.
مين؟
بيسان موجود. بيسان صارت موجودة.
موجودة بيسان. بس إنه مش قادر تتحدث يعني.
حولت لباناليست. حولت.
لا هي موجودين.
اوكي
موجودة دكتور موجودة.
يلا احكوا.
Can share your screen.
يلا بس شوي.
طيب. مع منا لوداد. وين وداد؟ وداد بأي اسم داخلي؟
وداد داخلي باسمين، اسم من اللابتوب واسمها العادي. اسمها وداد مرار.
طيب ما هي دائما يعني كمان واحد اوكي.
حسابين مش حساب واحد.
طيب هاي الاسم الثاني صار اوكي. هاي التنين دخلناهم وداد تو وداد.
دكتور اسمحي لها تعمل شير.
هي بتقدر. هلا تقدر تعمل شير هي. بتقدر تعمل شير وداد يلا.
تمام.
اوكي جود. Bisan with that, they are both of them, they are together.
Okay, you are from.
Yalla, go ahead.
وداد بيسان.
وداد مش ظابط يفتح معها المايك.
طب خلص هي انت.
لما هي احنا ظابطين الوضع اساس هي تبلش.
طب ما حدخلنا هلا هي اسم وداد هي. طب هي دخلناها احنا الاثنين وداد. اوكي هي انا اخر واحده. اوبا كم وداد عندنا.
So what do you think?
خلاص هاي جهزت يلا. هي الآن تقدر.
مين؟
وداد مع بسام.
وداد مرار؟
نعم.
هلا ضبط عندي.
Okay, وداد go ahead.
بس راح أعمل شير للسكرين.
نعم أعمل شير سكرين.
هيك ظهر.
نعم. Go ahead.
Good afternoon. Welcome to our project bidirectional PCR amplification of specific alleles by BASA for detection of virology zygosity in Palestine. My name is Widad Murrar and my partner is Bisan al-Fasfous, graduated students from Palestine Polytechnic University field Applied Biology. Our supervisor is Dr. Ruben Abu Ghazala. What we want to share with you today is a brief background about virology and BASA technique, our objectives, material and methodology, the results, and finally the conclusion. Many people think that mutations only lead to disorders and diseases, and a little know the benefits of some mutations. A specific autosomal dominant mutation found in the bone morphogenetic protein receptor type 1b, leading to an increase in the ovulation rate in sheep. This mutation called as Borola. The name refers to a property named Borola near Coma in Australia, where the first case of Borola was discovered in this farm. Bone morphogenetic protein receptor type 1b is one of fecundity genes located in chromosome 6 in sheep. This mutation happens in the locus, in the exon 8, in the base number 192 base pair. A substitution mutation from adenine to guanine happens in the base number 161 base pair, and causing a change from glutamine to arginine in the protein sequence, specifically in the amino acid number 305. As other dominant mutations, the zygotic state classified into three main categories: wild homozygous, mutant homozygous, and heterozygous.
669,843 sheep in West Bank in 2011, according to Palestinian Center Bureau of Statistics. Because of this large number, our project focused on sheep production due to their role in improving the economy. Also, the sheep offer the largest source of milk and meat in Palestine. The most common type of sheep in Palestine is local Awasi. The crossbreeding between local Awasi and East Frisian lead to the development of Asap sheep. Also, there is another two types of improved Awasi. One of them is Afik Awasi, which results from the crossbreeding between local Awasi and Borola merinos. This type is the main carrier for Borola mutation in Palestine. This statistic, which was done in many countries, including New Zealand, in New England, and Australia, show that Borola merinos have a higher ovulation ratio than pure breed merinos.
What is BASA? BASA refers to bidirectional PCR amplification of specific alleles, which is a rapid technique used to detect the zygosity of genes in one PCR reaction with using specific primers. And this technique hasn't yet been applied to detect the zygosity of Borola in the world. To distinguish between homozygous, wild or mutant, and the heterozygous in one PCR reaction, 4 primers are required: 2 outer primers and 2 inner primers. So in this reaction, we assume that outer primers are B and Q, the inner primers are A and B. So the gel electrolytic separation result will give it 3 bands with 3 different scents, and the genotype will appear as follows.
The main object of our project is to design a bidirectional PCR amplification of specific allele by BASA test to detect the zygosity of Corolla gene, which will help the farmer to know the genotype of their sheep in order to decide if they will keep the sheep for reproduction or for meat. Also, this test will be a substitution for Israel test because detection of the zygosity of Corolla gene is not done in the West Bank. The farmers go to occupied Palestine in order to do the test for their sheep. We decide to use a BIBASA instead of RFLB, standard PCR, and real-time PCR methods because BIBASA does not require a high cost or long time. Also, it's specific for SNPs and it's done in one reaction.
Material and methodology. Initially, the sequences of Polymorphisms protein receptor type 1b were found using Ensemble Genome Browser and NCBI BLAST. These sequences were aligned using GENIUS Prime Program and NCBI BLAST. This alignment helped us to find the mutant and what sequences, also to show if there is any other mutations in the sequences. As I said previously, the BIBASA technique requires 4 primers to it, 2 outer primers and 2 inner primers. So the outer primers were designed as in the standard PCR, while the inner primers were designed differently based on the mutation. The sequences contain adenine to guanine point mutation. So the reverse inner primer was designed to bind with the mutant allele, with the wild allele, with thymine complementary to adenine at the 3' end, while the forward inner primer was designed to bind only with the mutant allele with the guanine complementary to cytosine, also at the 3' end. After that, the 4 primers were tested using NCBI BLAST, NCBI Nucleotide, Genome Data Viewer, and also the Amplifix program.
The sample was collected from the farms. The blood was collected by resting the seed and inserted the needle into the jugular vein. After that, the blood replaced directed from syringe into edita tube and finally transported to the lab. For swab collection, a sterile swab cotton was inserted into the mouth of the sheep and then placed into plastic tube with a stabilizing solution, and after that, transport for the lab. In the DNA extraction step, Kygene kit was used. The extraction include many steps of incubation, centrifugation, and adding many reagents from the kit. For the sequencing, a specific PCR reaction was done in which only the forward outer primers and the reverse outer primers were used. The cycling condition shown here, and the gel was done for this reaction. The BQ band is the only band that was appear in all sample. This band was cut by using the scalpel for DNA purification. After that, the purified was amplified in a tube with total volume of 10.5 microliter, which containing the forward outer primer and other content from big dye exterminator kit of purification. Then the tube were put into PCR thermocycler, and after that, some solution and exterminator solution were added to this tube. Finally, tube content were placed into plates, and the plates was put into the sequencer.
The BIBASA reaction mixture was prepared by adding specific volumes of test reagents in the epigraphy tube. This mixture was used in the gradient PCR to determine the appropriate annealing temperature to give a clear band. So gradient PCR cycling conditions were the same as our BIBASA cycling conditions, but with different annealing temperature. Each row in the block of PCR thermocycler had a different temperature. So the range was determined from 50 to 57 Celsius to show the most suitable annealing temperature for primers after running the amplified DNA in the gel. After fixing the annealing temperature, the minimum optimal concentration of each primer was determined by dilution step. 0.4 micromolar was considered as a stock solution, while the other dilutions were as follows. The BIBASA cycling conditions were fixed as in this diagram with annealing temperature equal to 57 Celsius, and these conditions were used in the next steps. Here, a new reaction mixture was prepared, and 3 tubes were used in the amplification. Each one contained only 2 primers, and this step was done to ensure that all primers are worked well and can give each band separately. Finally, the quality of BIBASA test was determined by comparing its results with the sequencing results.
In the result, multiple sequence alignment was done for 16 sequence for Corolla gene from NCBI from different breed by using GENIUS Prime. The variable nucleotide is indication for Corolla mutation. All expected variant position within primer binding site were detected, and the frequency for each variant and its type were listed in this table. The position of primer was selected in which the forward outer primers and the reverse outer primers binding to intron 7 and 8 respectively, while the forward inner primers and the reverse inner primers binding to exon 8 around Borola mutation. At first trials, the inner primers were designed with a tail, which appear in the table with a red color, to increase its efficiency, but this tail caused primer dimer. So a second group of inner primer were designed, which appear in the table with a star, to solve this problem. The orange nucleotide is indication for the mismatch in the inner primers to increase its efficiency. This table shows length, GC content, and the degeneracy for the two groups of primers. The length of PQ band is the same in the two groups of primers, but the length of PP and AQ band is shown here. The best annealing temperature, which was determined by using gradient PCR, was 57 Celsius. All bands appear clearly in this annealing temperature.
As I said previously, each two primers were added together in the same tube to give each band separately. So the first gel contained the PQ band with length equal to 341 base pair. The second one contained the PP band with length equal to 235 base pair. The last one contained AQ band with length equal to 170 base pair. When the four primers are added together in the same tube, a competition happens between them. So the reverse inner primer can't bind specifically to give the PP band. Dilution step was done to solve this problem. At first, we make a dilution for the P primer, which doesn't give the PQ and PP bands. So we make the dilution for the Q primer, which gives us a hopeful result because the three bands appear in the second dilution, which equal 0.2 micromolar. After that, we make a dilution for the B and A primers separately, also for all primers together in the same tube. And the concentrations were fixed as in this box, 0.4 micromolar for primers B, and A, 0.2 micromolar for the Q primer. Here you can see our sequencing results for two samples from Sultan Farm. Both are wild homozygous with no mutant alleles. And here you can see a sequence from Summer Farm. It is a heterozygous sample with both adenine and guanine peaks. These sequences help us to confirm our bypass results. The zygosity of Borola was appeared like in this gels. The first one is contain a mutant homozygous with both PQ and AQ bands. The second one is a heterozygous with the 3 bands. And the last one is wild homozygous with PQ and a very faint PP band. Finally, we make a comparison between blood and swab samples. And as you see, blood is clearer due to a higher yield of DNA.
In the conclusion, Borola mutation has a good effect on the sheep because it increases the fecundity for the sheep. This gene requires a specific test to detect it. In this thesis, we design a test and test its ability to detect the zygosity of Borola mutation. This may help in improving other tests to detect the zygosity of other genes on the future. Thank you for listening, and now we are ready to receive your question.
Okay. One of the students is ready for your questions, colleagues.
Yes, leave questions to discussion, Dr. Khaled.
Okay, no problem. Thank you for both of you. And it was a very, very interesting, for me at least, the presentation. So we have Dalia. Are you ready, Dalia?
Yes, doctor, I am ready.
Okay, Dalia and 5 colleagues from the Coast University of Augusta Victoria Hospital, Bejala Hospital, and Leiden Hospital. Go ahead, Dalia, please.
Did the sharing work?
Okay, it's already shared.
Okay, great. Thank you for the introduction, doctor. I will present to you my thesis under the title of Microsatellite Instability and Mismatch Repair Genes Variation, Testing for the Identification of Lynch Syndrome Among Palestinian Colorectal Cancer in Palestine, supervised by Dr. Rania Busir. As we all know, colorectal cancer, like any other type of cancer, is an abnormal growth of the cells that affect the colon and rectum. It is caused by sporadic colorectal cancer and hereditary colorectal cancer, where the hereditary colorectal cancer is divided between familial, Lynch syndrome, and polyposis syndrome. Our focus today is on Lynch syndrome, which is the most common cause of hereditary colorectal cancer and account for 3 to 5% of all colorectal cancer cases. Lynch syndrome is caused by germline mutation in one of the mismatch repair genes, MLH1, MSH2, MSH6, PMS2, and EpCAM. It is inherited in an autosomal dominant pattern. Patients with Lynch syndrome have 50 to 80% risk of having or developing colorectal cancer throughout their lifetimes. Colorectal cancer patients with Lynch syndrome have some distinct characteristics that distinguish them from other colorectal cancer patients. These patients have an early onset of the disease. They tend to develop other extracolonic tumors. Their tumors are located in the proximal right side of the colon. Their tumors are poorly differentiated. They have high microsatellite instability. And directly because of the mutation in the MMR genes, they have loss of the MMR proteins in their tumor cells. In this study, we aim to describe the clinical and pathological characteristics of Palestinian colorectal cancer patients, to screen Palestinian colorectal cancer patients for Lynch syndrome, to mount a platform for the genetic defects associated with Lynch syndrome, to describe the clinical and pathological characteristics of Lynch syndrome patients in comparison to other colorectal cancer patients, and to study the pedigree of the patient identified with Lynch syndrome. To do so, we recruited 176 pathologically confirmed colorectal cancer patients from two of the most common or the most important hospitals in the West Bank that provide cancer treatment, Augusta Victoria Hospital in Jerusalem and Bejala Governmental Hospital. We filled a pathology questionnaire for all the patients that describe their disease characteristics. We retrieved formalin-fixed paraffin embedded blocks from the pathology archives from each hospital for all patients to be used in the immunohistochemistry and the microsatellite instability testing. And we collected data. For forming the family pedigrees. For the immunohistochemistry, we used two antibody screening panels using antibody for MSH6 and PMS2. We tested the expression of these two antibodies for all the patients. Then the patient with loss of expression in either one of the two antibodies, we tested the expression for the other two proteins, MLH1 and MSH2. We also tested 20 patients of the patient with intact expression. For the microsatellite instability testing, we did DNA extraction from both tumor and normal tissue for the patients. We used the DNA for touchdown PCR. We have 5 microsatellite markers. So we did a multiplex PCR for 4 of the markers and 1 singleplex PCR for the 5th one. And we did fragment analysis for the PCR products. Patients were divided between high microsatellite instability if they have 30% of or more of the 5 markers unstable, low microsatellite instability if only 1 marker was unstable, and stable microsatellite of all the 5 markers were stable. For the results now, the gender of our patients were 50-50, or the ratio was 1-to-1 between the males and females. The age at diagnosis ranged between 16 and 87 years with a mean age at diagnosis of 57.5 years. Patients were recruited from all over Palestine, including Gaza Strip, with the majority of our patients being from southern governorates, mainly Hebron, followed with Gaza and Bethlehem. The tumor of our patients were mainly located in the rectum, and only 12% of the patients had proximal tumors. Most of our patients had an advanced stage of the disease at diagnosis and at recruitment also. And 43 patients had stage 4 colorectal cancer, where the tumor have metastasized to several other organs. In some patients, we found the metastasis affected 4 other organs, and the main affected organ was the liver. In regard to the mismatch repair protein expression using immunohistochemistry, 12 patients had loss of expression of PMS2 and 1 patient had loss of expression of MSH6. These patients account for 7.4% of our patients. Among these patients, 54% had loss of expression of PMS2 and MLH1. 46% had loss of expression of PMS2, and 1 patient had loss of expression of MSH6 and MSH2. In this figure, we can see the immunohistochemistry for 1 patient who had intact expression of both MSH6 and PMS2. Whereas in this figure, we can see that this patient had loss of expression of both MSH2 and MSH6 and intact expression of MLH1 and PMS2. Whereas in this patient, there is intact expression of both MSH2 and MSH6 and loss of expression of MLH1 and PMS2. Among the patients with deficient mismatch repair system, these patients were mainly females. The age at diagnosis was similar to the other colorectal cancer patients. Here we can see the difference where the majority of these patients the location was divided between proximal and distal part of the colon, and only 1 patient had rectal tumor. The majority of the patients had moderately differentiated tumor, and only 3 patients had poorly differentiated tumor. And around 40% of the patients had stage 4 colorectal cancer. Microstability instability testing, we did the MSI testing for all the patients with deficient mismatch repair system and only 5 patients with the proficient mismatch repair system. Among the patients with the deficient mismatch repair system, 85% had high microstability instability, where 2 or more of the markers were unstable, and 2 patients had low microstability instability. And among the patients with a proficient mismatch repair system, 80% had stable microstability status. In this figure, we can see the fragment analysis for 1 patient who had high microstability instability. We can see the shift in the microstability status between the stable and the unstable alleles. We can see in BAT26 there is a gradient in the length of these microstability or these tandem repeats who ranged between 170 and 180 base pairs, whereas in NR27 it ranged between, or it shifted from 83 base pair to 73 base pair. In NR21 it shifted from 110 base pair to 99 base pair. In NR24 it shifted from 123 to 115, and in BAT25 it shifted from 151 to 140 base pair. Whereas in this fragment analysis, we can see the fragment analysis for 1 patient with a stable microstability status. We can see that the fragment analysis is identical or almost identical between the tumor and the normal tissues. This means that this is expected because the mismatch repair system is working and is efficient in these cells, and it repairs any error in the DNA replication. Whereas in this fragment analysis, we can see the distinct difference between the tumor and the normal tissues for the patient who had unstable markers. For all the 5 markers were unstable. We can also see in the tumor tissue that the tissue does not only have the unstable marker, they also has the stable markers, mainly because the tumors do not only have the tumor cells. In regard to the family pedigrees, we were able to interview 9 of the 13 families. 4 of them reported previous history of cancer. We can see in this pedigree that multiple family members had different types of cancer, whereas the other 5 families did not report any cancer cases. we can see the two pedigrees. The first pedigree show 4 generations of for the family of this patient who has been diagnosed at the age of 48. The second pedigree I wanted to add because this is very interesting. This is the youngest of among our patients. He was diagnosed at the age of 16, and none of his family members has any type of cancer. Limitation in our study, we underrepresented part of the northern parts of the country. The majority of our patients were from southern governorates. And the majority also of our patients had advanced stage of the disease, mainly because most of our patients were recruited from Augusta Victoria Hospital, where patients are usually referred for treatment and for surgeries. We suffered from lack of full medical records for our patients, so we were unable to test all of the variables and parameters we intended to use. And we were unable to confirm the diagnosis with Lynch syndrome. In conclusion, Lynch syndrome was estimated in 7.4% of the patients. The majority of these patients had loss of PMS2 and MLH1 expression. They were mainly females with similar age to other colorectal cancer patients and diagnosed at an advanced stage of the disease. And these patients with deficient mismatch repair system had high macrostability instability. Our recommendation is to test the patient with deficient mismatch repair system to determine the exact cause of loss of expression, to screen all newly diagnosed colorectal cancer patients for Lynch syndrome because it will affect their families and the treatment decisions. We recommend to early screen the family members for colorectal cancer patients. We hope to help in setting policies and guidelines that encourage family members to screen for colorectal cancer at the age of 20 or 25 every 1 to 2 years and yearly after the age of 40. Thank you.
Thank you. Thank you very much, Dalia.
You're welcome.
And her colleagues. It was a very big effort that they put on. Omar?
Stop sharing, Ms. Dalia.
Yeah, I did.
الخير داليا.
أهلا دكتور.
I have a question. Is high microsatellite instability means high disease progression and eventually increase the death rate?
Sorry, can you repeat? I did not hear well.
It's 5 o'clock.
I'm asking about you did the work on microsatellite instability, and when we found a high microsatellite instability, this means that there is a high progression of cancer in the patient, or at the end the death rate among patients with high microsatellite instability will increase?
The microsatellite instability is common among colorectal cancer patients, not specific to Lynch syndrome. I don't know if I'm understanding your question very well, but it's common among 15% of colorectal cancer patients. Usually, these patients had better prognosis compared to other patients. They are also, they can benefit from immunotherapy. Usually, patients with high microsatellite instability can use the immunotherapy. So I don't know if I answered or understood your question right.
Is there any correlation between high microsatellite instability and death rate?
No, or we did not test that, so I don't have any exact answer.
I see. Okay, thank you, Dalia.
You're welcome.
Dalia, can I ask you, just out of curiosity, one question, please? Why do you think that females has to suffer more than males? I mean, from such kind of cancer.
In everything, Dr. Khaled.
Yeah, we have bad luck, I think.
Not only in such obstacle.
So actually, my father
This
was.
is nature.
Yeah, yeah. I didn't mean that, but actually, my father is one of the people who died with this such kind of cancer, and a couple of his friends also. We know them that they are, but I actually cannot understand why. Did you go that so far on that? Why?
No, we only screened these patients, and among our patient, this is our finding. So no, we don't have.
So you are speaking about like statistically.
Yeah, only among our population or the study population.
Okay, just remind me how many?
176 patients.
Okay.
The ratio between them was 1:1, the original sample.
Okay. So how it ends up with the women with too much than the men if we began with 1:1?
I don't know. This is our finding. We don't have an exact cause. Also, we did not. We did only the screening. We did not confirm the exact cause or the exact mutation among these patients.
Okay, thank you very much.
You're welcome.
Did any one of our colleagues would like to ask Dalia anything?
دكتور هاشم كان رافع ايده.
Hashim?
Thank you very much. So I have 2 points here to make.
Yeah.
First one is probably the higher incidence in women has to do with hormones that usually tends to fluctuate during this period. But did you check for familial? Did you have among the patients multiple members of the same family or clan? You said something about doing the family pedigrees, and you have not mentioned it, or you haven't.
No, we did. I did mention it at the end. We did the family pedigree to expect if it's more hereditary, because as I said, the high microstability stability is not specific for Lynch syndrome. It is available in sporadic cases of colorectal cancer. So because we did not have the means to determine the exact cause of the mutations, so we said maybe the pedigrees will give us more hints maybe.
Okay.
So yes, we did the pedigrees. We were only able to interview or to contact 9 of the 13 families, and 4 of them, only 4 had members with cancers. The other 5 did not have any cancer cases among their family members.
Okay. So one other point. So microsatellite instability, I assume it would increase with the stage of the cancer. So the blocks, you determine the stage of the cancer from the blocks or from what? From the condition of the patients?
We did the DNA extraction from this, the formalin fixed paraffin embedded blocks.
No. You said that this number of individuals had stage 4 or stage 3.
Oh yeah, from the medical files. We returned to the medical files, and we saw the pathology reports of their surgeries.
And you got both controls. You got both the control sample and the cancer sample or the tumor sample from the same block, or you got it from what? From the veins? Like
no, the control was اوكي هحكيها بالعربي. عندنا المرضى البلوكس تبعونهم اخذوهم بعد ما عملوا عمليه الاستئصال، فبالعاده بيكون في مارجنز اللي لازم يكونوا فري اوف كانسر سيلز.
اوكي.
فاخذنا من المارجنز اللي هم النورمال تيشوز لكل بيشنت عشان تقدر اصلا هو تحدد لكل بيشنت اذا عنده مايكرو ساتلايت انستابيليتي لازم تقارن بين
Just wanted to make sure that they were taken at the same time, the same date.
بالضبط، هو العملية بنفس الوقت، في عندنا التيومر وعندنا النورمال.
اوكي.
فاحنا قارنا بين المريض نفسه، بين التيومر تبعه وبين النورمال تيشو تبعه. هلا في دراسات بتحكي لك ما في داعي انك تعمل هاي المقارنة، وانه هم ستيبل، يعني هدول المايكروستيلايت ستيبل بين كل العالم. بس احنا بمعظم مرضانا او كلهم تقريبا كان عندنا بنقدر نعمل comparison, so we compared their tissues.
In normal tissues, they should be stable.
Yes.
Otherwise, you could commit a crime and tomorrow your microsatellite will change and then you'll be innocent.
Exactly.
مبنية عليها. نعم.
بالضبط، فعشان هيك احنا حكينا الستيبل عندهم التيومر والنورمال كانوا ايدنتيكال، لانه هذا المفروض اللي يطلع معنا. على هذا الاساس احنا اصلا قررنا انهم ستيبل.
Okay, Dr. Hashim and Alia, we have to leave it here.
Yeah, thank you.
This discussion could go very, very far. And finally, could you please stop your share of your screen, Dalia? We go to the last presentation.
I did stop it long time ago.
Okay, no problem. Okay, beside structural and functional compatibilities of novel mutation, ASP July 420 Rs mutation in the factor X gene. Beside from the molecular genetics and genetic toxicology Arab American University Jenin. You are welcome, Usaid.
Thank you very much. Thank you for introducing me. It's a great pleasure to be here in the first version of this conference. Actually, the idea of this research came after colleagues of mine detected a novel mutation in factor 10 that leads to a severe factor 10 deficiency. So our idea was to understand the molecular pathology of this disorder. So I'm going in this presentation to start with a brief introduction about hemostasis and the coagulation cascade in particular, and then I'll move to our methodology and the results, and the conclusion after that. So hemostasis is the process that stops bleeding at specific sites of injury to stop blood loss. It consists of three major processes. It starts with blood vessel constriction or vasoconstriction to stop the blood flow at the site of the injury, and then there is platelet aggregation to form the primary hemostatic plug that will eventually stop the bleeding, and the coagulation cascade that ends into the formation of fibrin clot that supports the primary platelet plug. So in the coagulation cascade in this diagram we see it consists of the extrinsic pathway, the intrinsic pathway. The extrinsic pathway is the fast, or it's like the spark that starts the coagulation cascade. It produces few amounts of fibrin, but it also initiates the initiation of the intrinsic pathway. As we can see from this figure, that both the extrinsic and the intrinsic pathway joins at the factor 10. So from this we can understand the central role of factor 10 in the coagulation cascade. And factor 10 also then starts the common pathway that leads finally to the formation of fibrin molecules. So factor 10 as protein is a vitamin K dependent serine protease produced in the liver. It is activated by both the extrinsic and intrinsic kinase complexes, and it is a major component of the prothrombinase complex in the common pathway. This here is the polypeptide map of factor 10 protein. It shows the most important domains: GLA domain, EGF domains, the activation peptide. This site here is cleaved upon the activation of factor 10. We have here the catalytic domain or the catalytic triad. Both of the mutants in our study are located here near the catalytic domain or the catalytic triad. In this domain, it is called sodium binding domain. So factor 10 is also coded by factor 10 gene, which is located on the long arm of chromosome number 13, consisting of 8 exons and 7 introns. Okay. So any mutations in factor 10 gene leads to factor 10 deficiency or congenital factor 10 deficiency, which is inherited in an autosomal recessive manner. The general population incidence of factor 10 deficiency is 1 in a million, but our estimations also in the Palestinian population, it's six times the general population incidence. Factor 10 deficiency is classified into 2 types. The first type, we have a normal production of factor 10 with the produced proteins have reduced activity, while in type 2 there is, it's in type 1 there is decreased antigen and factor 10 activity, while in type 2 there is normal or near normal factor 10 production with affected activity. Until now, or up to date, about 135 mutations were reported in factor 10 gene. Most of these, or the major type, is missense mutation, as we can see in this table. So in factor 10 deficiency, the clinical presentation is increased bleeding or uncontrolled bleeding after blood vessel injury. We can see intracranial hemorrhage, increased bleeding after dental surgeries, bleeding in the joints, fatigues, ecchymosis, and so on. So in our study, we aim to understand the structural and functional implications of both of these mutants, the one we detected in the novel detected in a Palestinian patient, and another one was detected also, but was reported also previously, but they were very close to, in the same domain. And also to assess the capacity of synthesis and secretion of the wild type and the mutants compared to the wild type. First of all, we started with bioinformatic tools. We found out that both of the locations were conserved among multiple species. In bioinformatic tools, we use also Polyfen and Provian. Both of them predicted that this mutant and the other one were probably damaging and deleterious. This is a prediction. It is a brief prediction of the difference between the wild type and the mutant. And also the other mutant was probably damaging and deleterious. So we started in our experiments. We obtained this expression vector harboring factor 10 wild type gene. We introduced our mutations using site-directed mutagenesis using the following mutagenic primers. Then we transiently transfected our plasmids into human embryonic kidney stem cells, and cells were collected and lysed to measure the amount of intracellular factor 10. Culture media also was collected over the cells to measure the amount of secreted factor 10 and also the activity of this enzyme. We also extracted RNA from these cells to compare the expression between the two mutants and the wild type. Before we did that, we confirmed our mutagenesis using Sanger sequencing. So in the cell lysates, when we used Western blotting, we were able to detect factor 10 antigens in the cell lysate. and there was no significant difference. This was confirmed also by ELISA. There was no significant difference in the intracellular factor 10 in the cell lysates. In the culture media, also, we didn't also detect any significant difference of these mutants compared to the wild type. But the activity of factor 10 in the both of these mutants were decreased when we measured the functionality of these constructs compared to the wild type. There was no difference in the expression of these mutants compared to the wild type. We are now working on understanding the exact difference in the structure of both these mutants. We're using very advanced bioinformatic tools in collaboration, of course, with other colleagues from other institutes. We have now the data for the first mutant ready. We can see the difference in the structure due to the difference of charge between aspartic acid into asparagine. So to conclude, both of these mutants were thought to lead to local distortion of the specific domain they are located in. This leads to affecting factor 10 activity in both of these mutants. And that's it.
Okay, thank you very much, Usaid. Dear friends, do you have any questions for Usaid? Okay. Abdullah Abu Taha, go ahead. Abdullah.
Okay. Now, can you hear me?
Yeah, I allowed you to talk, Abdullah.
Okay, thank you so much.
Okay, most welcome.
Okay, Usaid, يعطيك العافية, very interesting work. The structures you showed at the end, were they just predictions using PyMol or bioinformatics tools, or these are structures based on the crystallography, etc.?
No, it's based on bioinformatic prediction or molecular dynamic simulations were done. We have collaborators now in an institute in Germany, Saarland, and with, of course, the Dean of Science at the Arab American University, Dr. Sabha.
Okay.
So
I got that. You know why? Because sometimes single point mutation can mess up the complete structure, so it doesn't change the domain as it shows in the bioinformatics. So what I wanted to tell you is just like to be really a bit careful with the interpretation, because changing the charge of a single amino acid can flip the whole domains. You know, it is not just the little active site which you speculate that this change by this picomole distance between this and this amino acid. So the whole structure might be messed up. This is the only thing I wanted to emphasize here. Thank you so much.
Thank you.
Okay.
Thank you very much for the end of the third session. We go back to Dr. Sharaf and Dr. Adir. Your mic's yours. Thank you.
Okay, we still have one, Mrs. Sabha.
Okay.
She's ready, or she is ready. She sent many messages a few minutes ago.
Okay.
Ms. Sabha, start your presentation.
Okay. Does it appear now?
It's okay. Okay. Go ahead.
Yeah.
And hurry up.
Okay. Okay. So my name is Sabha Rabaya. Today I will present my master's degree research work, where I studied the correlation of major iron metabolism related genes, single nucleotide polymorphisms, with autism spectrum disorder and a group of Palestinian patients. I did my work under the supervision of Professor Hisham Darwish at the Arab American University. I will start with a brief introduction about autism spectrum disorder, iron, and their relationship. How we formed our research hypotheses and research question, what are the methodologies we used, what are the results we got from our research, and then I will end up with a conclusion. So from the title, you notice that we talked about autism spectrum disorder and iron. So the question here is what makes us combine these two aspects? Okay. So to make that question clear, I will start with a brief introduction about ASD. It is a new group of neurodevelopmental disorders, including autism spectrum disorder and Asperger's syndrome, and many other disorders. The symptoms actually include problems in communication, behavior, and social interaction. And from these symptoms, you can see that they are hard to notice in children. So the diagnosis is considered late, from 1 year old to 3 years old. So this is one of the challenges with ASD, is that the problem of late diagnosis. So this has required the presence of genetic markers for early genetic diagnosis. And the next challenge with ASD is that the etiology and the pathophysiology is not fully understood until the moment. It has both genetic and non-genetic factors. So from these two challenges, we try to think about what are maybe possible factors that may contribute to ASD pathogenesis and may be used as genetic markers. We thought about iron for a lot of reasons. We know that iron is a very vital metal in our bodies. It is part of almost all oxidation reduction reactions. It is a cofactor for many enzymes. It is important for oxygen transport. In the brain, iron has vital rules. It is tightly and very tightly regulated, and this makes us think that any dysregulation of iron may affect brain development and many other aspects. And a very important point that researchers have documented that oxidative stress contributes to autism spectrum disorder pathogenesis, and we know that free iron is correlated with oxidative stress. And also, literature documented that genetic variations in iron genes affect iron homeostasis. So all these factors have formed our hypothesis that genetic variations in specific major iron metabolism related genes may be associated with autism spectrum disorder development. So to test our hypothesis, the first step was to decide what are the genes we're gonna test. So we chose 4 genes. The first one is the MT1, which is responsible for iron absorption in the intestines. The second one is ferroportin, which is the only known protein responsible for iron excretion in the blood circulation. The third one is an iron receptor, which is TFR1. And the last one is hepcidin, which is a key regulator of iron metabolism. So these are the 4 genes we decided to test. The next step was to decide what are the SNPs we're gonna study. To do that, we actually depended on literature review. We searched for SNPs that have been correlated with the neurodevelopmental or neurodegenerative disorders, specifically Alzheimer's disease or Parkinson's disease. So for the gene responsible for DMT1, we chose This is SNP RS224589. It is a SNP located in an intronic region, as you can see here. This SNP was correlated with Parkinson's disease. The next SNP in this gene is RS10482330. It is a SNP located in exonic region, but it does not result in amino acid substitution, and it is also correlated with Parkinson's disease. In TFRC gene, we chose this SNP, RS11915082. It is located in a promoter region. This SNP was correlated with age-related macular degeneration risk. In SLC40A1, which is responsible for reporting, we chose this SNP, RS1439816. It is located in an enteric region. It has been correlated with iron dysregulation and hemochromatosis, and also linked to Alzheimer's disease risk. In hepcidin, the responsible gene is HAM. We chose this SNP, RS10421768. It is also located in a promoter region. It is documented to be correlated with iron overload and dysregulation in thalassemia. So here are the genes and the SNPs we decided to test. So the next step, we recruited a group of 48 ASD patients and a group of control, 88 controls. We collected blood samples, we extracted DNA, and SNPs determination was carried out whether by DNA sequencing or fragment analysis, restriction fragment analysis. And then when I got the data, I did the statistical analysis using whether SPSS or another online tool called Cheeses. It is a very friendly use platform that you can enter your data and it will give you all the statistical association analysis. And finally, I did some bioinformatics analysis. So for the results, these are some representative images of the gels and the sequencing results. So actually, 3 SNPs gave significant correlation. For the correlation, I tested the association at the level of both alleles and genotype. For the SNP in TFRC gene, it gave significant correlation at the level of genotype and also allele. And the gene that's code for ferroportin, it also gives significant correlation at the level of alleles and also genotypes. And in the hand gene, it gives no significant correlation at the allele level, but gave significant correlation at the genotype level. So these are the 3 SNPs that gave significant correlation. Ferroportin was the most significant one. So next to that, I did association analysis between each SNP and some key symptoms, such as self-behavior, language skills, self-expression. 2 SNPs actually gave significant correlation. The SNP in DNT1, which, if you remember, gave no association at the level of the whole phenotype, but gave significant correlation with self-expression. And also, the SNP and ferroportin gave significant correlation with self-behavior. So the final step was that I tried to predict or to see what may be the effect that these SNPs do to be correlated with ASD. And as I mentioned, these SNPs were located in intronic region. So I used specific bioinformatics tools that are specific for SNPs located in intronic regions. For the SNP and TFRC, a rank of 4 in Regulum DB means that there is an evidence that this SNP has a significant regulatory role. It also affects the binding.
Try to finish.
I'm finishing.
Take over time.
Okay. So it also affects the binding of some transcription factors. The same for ferroportin. And also for the hemp gene, it also gives a rank of 2B, which means that there is a strong evidence that this SNP has regulatory effect. So as a conclusion, this study is the first study to try to draw the attention toward the possible role of iron dysregulation in ASD pathogenesis. We documented that some polymorphisms in 3 major iron metabolism genes may be correlated with ASD pathogenesis, and these may be used as genetic markers for diagnosis. I recommend that this study made on a larger cohort of patients and controls, and maybe other genes and SNPs can be included, and maybe include some hematological parameters such as iron levels. Thank you for your attention, and if you have any questions.
Thank you, Rabaya. If you have any questions, just raise your hands, please.
Fine. If anybody has questions, you can ask. We are now in the general discussion. If anyone would like to ask something to any of the speakers in these sessions, he can ask. طيب، على كل الأحوال يعني إحنا سعيدين جدا بالسيشنز اللي قدمناها في هذا اليوم، في اليوم الأول أو المؤتمر، يعني المؤتمر الأول لتجمع العلوم الحياتية، البيولوجيا التكاملية في والتنوع من أجل الاستدامة والاقتصاد الحيوي. بنشكر جميع اللي شاركوا معنا، يعني حقيقة كانت يعني مشاركات قيمة، يعني سواء في المايكروبيولوجي لما تحدثنا about the harsh environments, how to induce immune system of the microorganism to be adapted for the harsh environments, either for electrolytes inducing, or to the next sessions about the plant biology, how we can envisagement the solar radiation or the climates to produce biomass of microorganisms to be used as biofertilizers, even to screen for higher plants which are resistant for such stresses, and the genetics applications also, how to use or to control some disease, human disease such as cancer, breast cancer, and etc. So we hope to meet tomorrow once again, and the general discussion with the closing remarks will be for tomorrow. Okay. For now, if there's no questions or any comments from the keynote speakers or the panelists or anybody else,
I just want to say thank you for including me in this, and I'm truly looking forward to more collaborations. And I was proud of all the students and of all the research that is taking place in Palestine. وإن شاء الله للأمام وأكثر وأكثر ويكون في عنا productivity لأنكم أنتم كلكم ما شاء الله insistent على أنكم تعملوا research قيم وتشاركوا فيه العالم مع كل الظروف اللي احنا فيها. شكرا.
شكرا لك دكتورة نهال. هذا ما نطمح له احنا يعني، واحد اهدافنا يعني في هالمؤتمر انه نقدر نعمل تشبيك ونتوركينج with scientists overall the world يعني. So ان شاء الله we have to get the ability to do such difficult job. اوكي. طيب انا يعني بدوري بدي اشكر الجميع على المشاركه في المؤتمر السنوي الاول، وفي اختتام اعمال اليوم الاول للمؤتمر نتوجه بالشكر الجزيل إلى جميع المشاركين من الكينوت سبيكرز، الدكتورة نيهال، الدكتورة كريستينا، الدكتورة حنان ملكاوي، اللي شاركونا اليوم وعطلوا أوقاتهم ومشاغلهم وشاركونا مؤتمرنا اليوم. شكرا لكم على وجودكم معنا وعلى المعلومات القيمة اللي أثرت يعني مؤتمرنا إلى اليوم. بنشكر جميع المشاركين الباحثين والباحثات الذين قدموا أوراقهم العلمية في هذا المؤتمر في هذا اليوم لهذا المؤتمر، وبنشكر جميع الطاقم يعني القائمين على المؤتمر، اللجنة العلمية واللجنة التحضيرية ومدراء الجلسات جميعا. وبنشكركم كلكم، الله يعطيكم ألف عافية، كفيتوا ووفيتوا، وشكرا لحضوركم ولوقتكم ولتعبكم ولكل شيء. يعني مش عارف كيف بدي أشكركم أكثر من هيك، يعني من كل قلبي بشكر الجميع على هاي المساهمات وعلى هاي يعني المشاركات، وإن شاء الله السنة القادمة بتكون مشاركات أكثر من هيك وممكن ومؤتمر أفضل من هيك، وإن شاء الله بتعمل فائدة على الجميع حسب الأهداف المرسومة، إنك تجمع فلسطيني للعلوم الحياتية، وبيكون مظلة التجمع لكل البيولوجيين الفلسطينيين في الداخل والخارج والأوفر أول ذا وورد كمان تحت هذه المظلية. وإحنا إيدنا ممدودة مفتوحة للجميع، إن شاء الله نحط إيدنا بإيد بعض ونتعاون مع بعض ونعمل شيء للأجيال القادمة، لأولادنا مستقبلنا ولبلدنا وطننا، وإن شاء الله إحنا نقدر نقدم شيء في سبيل هذا الوطن.
دكتور شرف إذا بتسمح لي، طبعا بنتشرف بالناس اللي كان عندهم توكس اليوم وبالاتندنز الاتنديز انهم يشاركونا كمان بكره، يشاركونا في محاضرات اللي حيتم القائها مما يسري يعني المؤتمر، فبتشرفونا كمان بكره مش بس انه اليوم. يعني كثير بكون جيد مشاركتكم.
شيء جميل، وبكره راح يكون عندنا يعني الفقرات الحاسمه خلينا نسميها، يعني لانه بكره عندنا
بكره الجوائز صح؟
الجوائز نعم فالاشياء كثيره بكره ان شاء الله يعني. فبالعكس احنا بدنا بكره نسمع من الجميع، الكل يكون موجود بكره، نسمع من الجميع وناخذ منهم فيدباك، ويعني لحتى للمرات القادمه يكون في عندنا امبروفمنت بشكل افضل للمؤتمرات العلميه والتجمعات تبعاتنا ان شاء رب العالمين. إذا في حد حابب يضيف شيء قبل ما ننهي أعمال المؤتمر لهذا اليوم. دكتور أيمن مش موجود، دكتور
اعتذر بعد في إشي صار عنده بالنت.